Hello everyone, my name is Marin Patel and I am here with a new video about how plants reproduce and what are the products of this process. I am here with my two classmates Ali and Akhilesh to complete this video, and I am going to introduce you some interesting things about the structure of anther. Maybe you don't. Let's start with the structure of stamen. So as we all know that androecium is the male reproductive part of flowering plant. Then the structure of stamen contains anther and filament. Anther, as you can see that anther is connected with the connective. It connects both the lobes of anther. So let's see the structure of anther. So the structure of anther contains two lobes. Therefore, the anther is also known as bilobed or diathecous, and each lobe contains two chamber of pollen grains. Therefore, there are total 4 chambers. Therefore, it has also characteristic of tetrasporangiate. Each chamber contain lots of MMC, which are microspore mother cell. They are the homogeneous cell together form sporogenous tissue. Further, MMC will divide 4 times by meiosis to produce pollen. So, as you can see in this picture, there are 2 lobes and 4 chambers of pollen grains, and each chamber contains the MMC which is diploid, and at the maturity of anther, both the lobes are fused together and form one theca. Now we will talk about the layers of anther. There are 4 layers of anther. First is, and the outermost layer is epidermis, and second is endothecium, and third, which contain multiple layers, is middle layer, and then fourth and the innermost layer is tapetum. So as you can see in this figure, there are 4 layers. I drew it, and first layer is epidermis, second layer is endothecium, and these multiple layers which cover the inner part of lobe is middle layer, and the last one and the innermost layer is tapetum. Now we will talk about the functions of all the 4 layer. So as you can see in this figure, epidermis and endothecium both are protective in nature, and the last one and the innermost layer tapetum have 2 functions. First one is the nourishment of a microspore mother cell which is diploid, and the second one is the secretory nature which secretes the sporulin, which form the outermost layer of the pollen grain, which is known as exine. We see the structure of pollen grain and the structure and function of exine, which is formed by sporopollenin. So you can see the structure of pollen grain. The outermost layer, which is discontinuous in nature, is known as exine. There are gap between these 2 layers is known as germ pore, and inside the exine there is a continuous layer which is known as intine, and inside this intine there are 2 cells, which is one is larger one and one is smaller one. Larger one is known as vegetative cell, and the smaller one is known as generative cell. Larger one, which is vegetative cell, is also known as tube cell. Basically, it forms a pollen tube from stigma to the ovule, and the smaller one, which is generative cell, it forms 2 male gametes.
Now comes female reproductive part, which is consist of stigma, style, and ovary. Many ovule is present in ovary. In ovary, ovule is connected to placenta through funicle. If funicle is present, it is known as funiculated, and if funicle is absent, it is known as sessile. Here, ovule is connected to placenta through funicle. If this funicle is present, it is known as funiculated, otherwise it is called as sessile. Ovule contains one megasporangium, also known as nucellus. Many megaspore mothers is present in nucellus, and it forms sporogenous tissue. The opening through which male gamete enter the ovule is known as micropyle, and base of the ovule is known as chalaza. At this point, male gametes enter ovule, and it is known as micropyle, and base of this ovule is known as chalaza. Megasporangium is surrounded by 2 protective layers. The outer one is known as outer integument, and inner one is known as inner integument. There are 3 ways through which ovule can connect to funicle. The first one is known as orthotropous, in which micropyle, chalaza, and funicle forms a straight line. Ovule makes 0-degree angle with funicle. Second one is known as anatropous, in which ovule makes 180-degree with funicle, and third one is known as hemitropous, in which ovule makes 90-degree with funicle. Micropyle, chalaza, and funicle are in a straight line, and it makes 0-degree angle, and it is known as orthotropous. Here, it makes a 180 degree ovule makes 180-degree with funicle and it is known as anatropous. Here, ovule makes 90-degree with funicle and it is known as hemitropous. Here, many megaspore mother cell is present, but which is at micropylar end will undergo meiosis, and by meiosis it will form 4 megaspore. And out of this 4, this one is towards the chalazial end, and it will undergo 3 times mitosis, and other 3 will degenerate. From first mitosis, it will form 2 cell, and from second mitosis, it will form 4 cell, and from third mitosis, it will form 8 cell. 4 towards the chalazial end and 4 towards the micropylar end. 1 cell from chalazial end and 1 cell from micropylar end come in center and fuse together, and they will form polar nuclei. 3 cell remaining at chalazial end is known as antipodals. This 3 cell is known as antipodals. 3 cell also remains at micropylar end, and middle one is known as egg cell, and so the 2 cell which surrounds this egg cell is known as synergids. Now egg cell will fuse with male gamete and it will form zygote, and polar nuclei will fuse with male gamete and it will form endosperm.
Now we are going to see the important sections of this chapter, that is double fertilization and post fertilization, and how male gametes reaches to the ovule to fuse with egg cell and secondary nucleus to form zygote and endosperm. By pollination, pollen grain reaches to the stigma, and pollen grain has 2 cells, one of which is vegetative cell, which is also known as tube cell, and another is generative cell. Vegetative cell, with the help of the stigmatic secretion, forms pollen tube, which reaches to the ovule. Pollen tube carries 2 male gametes, which are released by the generative cell, and 1 tube cell, tube nucleus, which is released by the vegetative cell or tube cell. Now we will see the 3 ways by which the pollen tube will reach to the embryo sac and it will release male gamete into it. The 3 ways are porogamy, mesogamy, and chalazogamy. Entry of pollen tube into embryo sac can only be done through synergids. That means the pollen tube will enter into the embryo sac only through these 2 synergids. Now, how pollen tube will reach to the embryo sac? There are 3 ways. One of it is porogamy. In porogamy, the pollen tube enters into the embryo sac by the micropylar end. By the micropylar end, it travels and reaches to the embryo sac. The pollen tube contains 2 male gametes and 1 tube nucleus. The tube nucleus degenerates as the pollen tube reaches to the embryo sac. Now, we will see the second way by which pollen tube will reach to the embryo sac, by mesogamy. In mesogamy, the pollen tube travels to outer and inner integument to reach the embryo sac. Now the third mode by which pollen tube reaches to the embryo sac, that is by chalazogamy. In chalazogamy, the pollen tube comes through the chalazial end and reaches to the embryo sac. The transportation of the non-motile male gamete to the non-motile female gamete through pollen tube is known as sphenogamy. As the pollen tube enter into the embryo sac through synergids, it starts absorbing the protoplasm of the synergids and gets swollen. By swelling, it gets burst out and releases 2 male gametes. One of the male gamete fuses with the egg cell, this is known as syngamy, and it forms zygote. And another male gamete fuses with the secondary nucleus to form primary endosperm nucleus. This is known as triple fusion. As the secondary nucleus is diploid and the male gamete is haploid, they form triploid. Thus, the fusion occurs twice. Therefore, it is known as double fertilization. After fertilization, the 3 antipodals and the 2 synergids get degenerates. Now the event that occurs after fertilization, that is post-fertilization. After fertilization, the zygote develops into embryo and the primary endosperm cell along with its primary endosperm nucleus forms the endosperm. The endosperm gives nourishment to the embryo until it becomes seeds.